It's my honor to introduce Dr. Ayesha Mahmud. She's an associate professor in the Department of Demography, University of California, Berkeley. Prior to joining Berkeley, she was a Rockefeller Foundation Planetary Health Fellow at Harvard University. She earned her PhD in demography from Princeton University in 2017. Her research at the intersection of population dynamics, environmental change, and infectious disease ecology. Integrates statistical approaches with biologically mechanistic models to understand how demographic processes, human mobility, and environmental variability shape the timing, spatial spread, and burden of infectious diseases. Drawing on diverse data sources, including disease surveillance systems, hospital reports, climate models, and population service, she investigates disease dynamics across regions in Asia, Africa, and Americas. Let's welcome her for her talk. In just a second. Thank you so much. And thank you so much, Betsy, for organizing this amazing symposium. It's a real honor to be here, and I'm sad this is ending. And I hope someone carries it on. What a great symposium. I had a really great time yesterday meeting everyone, and this is a really great format.
Yeah, I don't know how to get rid of it.
So on my slide there.
Yeah, because it's overlapping.
Please downloading my slide. So I'm a demographer, but my training is kind of at the intersection of ecology. So my PhD advisors were both ecologists, and so I kind of straddle this world between ecology and social sciences and demographics. And so a lot of my work kind of is trying to bring together that interdisciplinary perspective to think about infectious disease transmission. I'll wait 1 second.
Sorry. Yeah, right.
The last one where we have the most technical difficulties.
Okay, and so we get back to where their slides are. Right here. No, where are their slides? No, I'm fine. No, yours were. Yeah, so I'm gonna airdrop over here. Okay.
That one, or the top one, or the second?
It's the PDF will probably be the easiest without
Okay,
issues.
Here we go. This one.
Mm-hmm.
There we go. Wait, come on, go. Maybe. There we go. Is it showing up? You have to say accept it. Did you? Yes, there it goes. Just 1 minute, and then you can open it. There you go.
No, you full screen, enter full screen.
Wonder if I can get this to show on the other one. Actually, it's probably fine then.
Yeah,
Let
so
me
just
yeah.
minimize this. Or you
Yeah.
want to do that? Just minimize this.
If you stop sharing and then reshare.
Yeah, actually, if you
Yeah,
could
it's
just, you
fine.
don't want to stop sharing. You don't want to mess up. So where is it? Is it?
It's in this desktop.
It's in the desktop. Okay.
We just saw it.
Yeah, I can.
You got it. You
Yeah.
got it. Yeah, because we want to. Yeah, there you go. There you go.
Okay.
But it's there.
Oh
No,
my
it's,
God.
but so you have to close the other one.
Okay.
You have to just minimize the other one. So it's using. So you can close that one altogether.
Oh, nothing's showing up there.
Yeah, see, it's not sharing the desktop.
I think it's just sharing the.
Yeah, it's sharing this screen. Sorry, which application
That's
is
not
it on?
her. That's not her.
Yeah.
You go to the Zoom window and you.
So it's sharing this on Zoom. The problem is it's projecting that right now. Let me stop extending. Okay.
Choose mirror from the menu.
Uh,
You have to go choose mirror from the menu.
choose content.
We got both of the tech people here.
There, you got it. You got it.
Yeah.
Yeah.
Sorry.
And you just slide that. Yeah, just slide that over with your.
Okay.
Can I move
and
this?
then yeah, let's.
That,
And then
like
just
that?
say, okay.
Here, it's gonna block, yeah, if we minimize.
Um,
Yeah, we don't
anyways,
need to see it.
Sorry.
Thank you so much. Okay, so I'm again really delighted to be here, and so this is the realm of infectious disease that I'm interested in. This is a pretty large scale population level view. So I've worked with microbiologists and people who take a more zoomed in view, and I think it's kind of interesting to think about, at least in a setting like this, what are the different perspectives you can get from each of these views. So in the world I live in, I think about infectious diseases very much in terms of what's happening at the population level. And so you can think about, for example, an infected person getting introduced into a population. Over time, you might see the infection spread, and then maybe you see some recovery. So this is a very stylized schematic of what happens. And then the type of data that I often work with is things like this outbreak curve. So this is data from public health systems and surveillance data. You're only probably capturing a fraction of infected. So I think a lot about statistical estimation and what we can learn from these kinds of data. And the typical types of questions I'm interested in are really to do with what are the factors that drive the sort of dynamics that we see at the population level. So I think a lot about human demography and the ways in which demographic changes and demographic factors affect these outbreak dynamics. So when we think about who is at risk for a pathogen, demographic processes are really important. So births add to the pool of susceptible people who can be infected. And so, for example, if there's changes in timing of births, birth seasonality, all of these have been shown to affect the dynamics. I also work a little bit on migration and population mobility and thinking about meta-population dynamics. So if you have sources and sinks of pathogens, we might want to think about how people are moving between those sources and sink populations. And then what I mostly talk about today is thinking about the climate aspect of this. So I've been doing some work thinking about what are the both climatic and environmental drivers of infectious disease dynamics. How do they affect the timing of outbreaks that we see in populations? How do they affect the spatial patterns of outbreaks that we see in populations? And thinking a little bit about how do we estimate these kinds of relationships. And then most recently, what I've been doing is thinking about kind of not just demographic and climatic factors, but also the social and political and economic conditions in which these outbreaks happen. And so thinking about things like political polarization for vaccine uptake, thinking about socioeconomic differences and who's at risk and of exposure, but also who's at risk of dying from infectious diseases, and so on. And all of these things are jointly shaping the outbreak that we end up seeing in population. And so a lot of my work is trying to think about what are these both extrinsic and intrinsic drivers. How do we statistically estimate these relationships? It turns out to be really hard because we often have, the data we have only looks like the sort of in some ways, the tip of the iceberg. We see the severe cases and we kind of see our outbreak curve, but we don't really often have granular information on the mechanisms that's leading to that process. So a lot of my work is trying to think about how do we statistically estimate these drivers. And then if we are able to statistically estimate a relationship, for example, between a climate variable and transmission, then how do we use that in a mechanistic modeling framework? And so that's really more sort of work on my ecology hat, which is thinking about very simple mathematical models of transmission. So I typically work with compartmental models, the classic models that I think many people in this room have worked with. So thinking about these susceptible infected SIR compartmental models, which I'll show you an example of that work. And then what we've been trying to do sort of in the last 5 or so years is think about if we are able to have these mechanistic models that tell us something about transmission dynamics, and we can use these models to simulate what will happen if there are changes in any of these factors. And so we've been thinking about what are the implications of both demographic change and climate change for the future of pathogens. I should say I'm also very happy to take questions as I go along, so if people have questions, please just stop me. So here are some of the factors that I've been thinking about, and definitely I haven't thought deeply about all these factors, but these are some of the ways we think climate might be affecting population levels and dynamics. So we can think about climate affecting the range for reservoir species. We might also think about how climate is changing the ways in which animals and humans interact and the risk of pathogen spillover. There's also evidence, and I'll show a little bit of this, of how climate might be affecting both the transmission of pathogens and the susceptibility of humans to infectious diseases. And then climate also affects the geographical range of vectors, and it's particularly important for things like mosquito-borne illnesses, which I think hopefully we'll hear some of that from Courtney later on. And then at the same time, these climate changes are happening. We also see pretty large scale demographic changes happening. So we can think about the ways in which land use has been changing over the past century, pretty rapid urbanization and population aging. So we've seen falling birth rates and aging populations, and I'll talk about that kind of towards the end of the talk. And there's many ways in which these demographic changes can affect disease burden. I'm not going to go through all of this, but the thing that I'll focus on today is thinking about how aging might result in possibly a larger burden for things like influenza in the coming, say, 50 years. So when we think about the impact of climate on pathogens, there's a recent review paper in Nature Climate Change looking at the published literature on this topic, and they say that in their systematic review, they find that more than half of known infectious diseases have some association with the climate hazards. And so the vast majority of these studies have looked at vector-borne diseases, which makes a lot of sense because the vector biology is often very strongly linked with environmental and climatic conditions. But also waterborne pathogens, so thinking about diarrheal diseases. And I think we might hear both about vector-borne diseases and waterborne pathogens today. So there's not just the ways in which we might think weather variables might affect infectious diseases. There's also, with climate change, we've seen an increase in extreme weather events and natural hazards. So there's things not just temperature and precipitation, but things like floods and droughts and storms that have been shown to have an impact on these diseases. So today I'll focus on respiratory pathogens and airborne respiratory pathogens, which I think have been less studied in the climate literature on this, and probably with good reason. I think that we have many biological reasons to think that vector-borne and waterborne pathogens will be the most impacted. But I'm going to show you some results to show why we might want to think about climate for airborne pathogens as well. So in the first part of this, I'm going to talk a little bit about how climate affects flu dynamics. I've been working on flu in the last 5 years and thinking about it from many different ways. So the first part, I'll talk about some work we've been doing to think about the climate impacts, and then I'll talk about a little bit about what we think changing demography and age structures might mean for flu burden. So I think probably most people are familiar with this, but just a very quick overview. There's been some work in the past 10 years or so trying to establish this link between climate conditions and influenza transmission. So when we think about typical flu outbreaks in temperate regions, they tend to happen in the dry, cold winter season. So in the US, there's some variability in timing spatially, but most of the time we see flu outbreaks happening roughly around January, February, and it's kind of the driest, coldest months in the year. And there's a lot of evidence now from both lab studies and these population levels of epidemiological studies that show that low humidity and low temperature is beneficial for influenza transmission. And so this is some figures from work by Jeff Shannon and colleagues who show this relationship between specific humidity on the x-axis, and then on the y-axis on the left, we have percentage transmission, and then the percentage viable for the virus. And you can see this sort of downward relationship where as humidity increases, transmission goes down. So the highest transmission both from lab and publication studies, we see that highest transmission tends to occur in temperate regions in these dry seasons. This has also been shown in sort of lab settings. So Anise Lowen and colleagues did some really beautiful studies with guinea pigs that were in separate cages, and they varied the humidity and temperature conditions in the lab and looked at the influenza transmission between the guinea pigs, which is really nice to have that evidence, because when we think about this, and often when I present this link, the first thing that comes to people's mind is, well, is there something about whether that's affecting the virus, or is it really that humans behave differently in different kinds of weather conditions? And I think we do have some evidence that humans do behave differently. So in another line of work, I spent a lot of time measuring human contact networks to think about how these networks change over space and time and what these networks mean for diseases. And so we do see some evidence that contact networks do look different at different times of the year. But I think the lab studies give us some evidence that it's not just the human behavioral side that's affecting it. There's also other aspects of the transmission dynamics that are being affected by climate. And this is happening through how climate variables are affecting virus survival and the viability of the virus. So this is what we've seen in temperate regions. When we look at data from tropical regions, it's a little bit more complicated, the story. So in tropical and subtropical regions, the seasonality of influenza is first of all less well studied, because we often don't have data from these countries to look at long-term seasonal dynamics. So most of low and middle income countries don't have national influenza surveillance systems. So there's often not really great data for studying this. But for the countries where we do have data, what folks have seen, and this is a figure from Tamarius and colleagues again from Jeff Shaman's group, is that when countries tend to have a rainy season in the tropics, we see influenza peaking roughly in the rainy season. So it's no longer a winter disease in the tropics. You might often see it in the hot, humid months, often following a rainy season. And then in a lot of countries, influenza we don't actually see any evidence of seasonality. Influenza is happening year round, and there's no clear seasonal pattern that shows up. So in tropical countries, the story is a little bit less clear. There's also not really great lab evidence for what happens once humidity goes past a certain range. And so in this figure, the authors suggest that perhaps there's a U shape where transmission is highest in these dry, very dry conditions, and then perhaps transmission is also high in these more humid conditions, right? Unfortunately, we actually don't have great data for what's happening on this other side of the curve, but one of the things my collaborators and I wanted to do as well, can population level data tell us something about this?
What if they hypothesized might be happening on the other side?
Yeah, that's a great question. So in their paper, they go through a few different things. One is this human behavioral aspect. So in the really humid, perhaps if the humidity is correlated with it being the rainy season, people are indoors and behaving differently. So that's one hypothesis. There's a really great new paper out by Jamie Lloyd-Smith and his group looking at enveloped viruses. And so they don't look at who they were really interested in SARS-CoV-2, but showing some evidence that high humidity might actually increase transmission by affecting the virus in ways that my biological knowledge will fail me. But the E-Life paper by Jamie Lloyd-Smith has some more biological mechanisms for what it could be. Yeah.
Are these species dependent? Not viral species dependent?
Yeah, that's a great question. So I think that these results I think do not distinguish between different strains of the flu. There is a paper by Cécile Vivout that tried to look at what was the impact of climate on different strains, like influenza B versus influenza A strains, for example. And from what I remember, I don't think they found a huge difference, but they also argue in the paper that often we don't have really great data on strain-specific incidents at the population level to estimate this. I think more and more of that is becoming available, so there's potential to look at it, but I don't know if we have a great answer for that. Yeah. Other questions? Okay. Okay, so we set out to try and look at this question in the context of Bangladesh. So in Bangladesh, influenza is seasonal, and the peaks happen during the monsoon season. And there's been previous work that has shown that there's a spatial gradient in the timing of outbreaks. And so one of the questions that we wanted to know is, can we explain the seasonality of flu? Does climate seem to be associated with the seasonality? And then if it is associated, and we think that there's going to be changes in humidity and precipitation and temperature in the future, how might that affect the future burden? So this is joint work with Rachel Baker, who is a faculty at Brown. She's a climate scientist. And then Pamela Martinez, who is faculty at UIUC, who's an ecologist. And so we kind of put our heads together to think about how could we try and understand this question. So in Bangladesh, there is some influenza surveillance. So these are sentinel surveillance. So these come from sentinel hospitals around the country. And we have data from 12 of these locations, thanks to our collaborators in Bangladesh. And we can look at not quite the burden of flu, but we can look at what proportion of the tests that they do in these sentinel sites turn up positive for flu. And so this gives us a sense of not the overall burden, but the kind of seasonal patterns of flu in Bangladesh. And so this data is showing the cases in green. So these are the number that test positive, and then the blue is showing the proportion of tests that are showing up positive. So you can see that it is a pretty seasonal disease. It's not peaking in the winter, which is what we would see in temperate regions, but it's peaking in sort of the kind of middle of the year, which is the monsoon season in Bangladesh. And so we broke this down by season. So here I'm showing the, on the x-axis is temperature, on the y-axis is humidity, and then the colors are telling me how the percentage of my tests that are showing up positive for flu. So the darker the color, a greater proportion of my tests are showing up positive. And so this figure is showing a few different things. One is it's basically showing us what's the temperature and humidity range that we see in these seasons in Bangladesh. And so each dot is from a sentinel site for a given month. And you can see that most of the cases are happening in the monsoon, which tends to be the hot and humid time period. So the dots are all clustered together. So it's a pretty narrow range of humidity, but it's really high, and the temperature is pretty high as well. And then we see some cases in the pre-monsoon season. So this would be April, May. And you can see that when we do see cases in the pre-monsoon season, they also tend to be in the months where, or in our observation, the months where the humidity was and the temperature were also pretty high. And then in the winter, there's actually very little cases. This is when the temperature and humidity are both at the lowest point. And we see some cases, but in general, the proportion of test positive in winter is quite low. So we fit some statistical models to this to try and capture some of this variation. And you can see that, if we look at month, for example, that's probably the most clearest. Flu is peaking typically in, the kind of monsoon season, which is roughly May, June, July, August sometimes. So the monsoon season is also shifting, which is an interesting question to think about. And then if we look at the relationship we estimate, we do kind of see this bimodal relationship. I should point out that we have no observations for humidity below, I think, 4, so that's probably the lowest. So the last bit is just extrapolating from 5 onwards. So you can see those confidence intervals are huge. But we're pretty confident in kind of this shape we see on the other side. And then we see some temperature effect. The confidence intervals, yeah, it's huge because the temperature range in Bangladesh is so narrow. There's not really enough variability to really pick up a strong effect. And then we see some effect of precipitation, but it seems to be most of it is being captured by humidity. So we took this and we said, okay, can we put this into a classic sort of compartmental framework to try and simulate the dynamics? So we just took these very simple SIR frameworks. We have susceptible, infected, recovered. We modified it to adapt it for flu, so there's waning immunities. Once you recover, you can go back to your susceptible compartment. And you can see from the equations, it's pretty simple model, which to me is, allows us to think about a little bit more carefully about, well, if the simple model can capture some of the dynamics, maybe we can learn something from it. So there's many other things that affect the dynamics that are not going in here. But what is going in here is climate variability. So we have our transmission rate, which is the beta here, and we have the transmission rate dependent on humidity. So this comes from the relationship we estimated using our statistical model, and then we can use that statistical model to estimate what the relationship of humidity and in this case I'm showing R naught, but it's really beta that it's affecting. So this tells me how many, so R naught would tell me how many new cases I would get in a fully susceptible population if I introduce one infected person. So it's telling me something about how quickly my outbreak will grow. And so I can make my transmission be dependent based on this bimodal relationship we estimate. And then we can simulate our model and run it forward. So here I'm showing the observed data in green for 4 different locations in Bangladesh. And then the yellow is the simulated data with some uncertainty bounds around them. And you can see that even this pretty simple model is obviously not perfect. We're missing a lot of the kind of magnitude variation, but we do pretty well in capturing the seasonality, which is remarkable because the only thing we've put in this model is really this humidity and a slight temperature dependence. So mostly it's humidity driving it. I think this is, we're very cautious in sort of interpreting these kinds of models. We don't think this tells us absolutely that humidity is what's driving it. But when we run these models by putting in other things, such as other kinds of seasonal forcing, we see that the humidity model does a lot better. So we test it against other kinds of models to try and convince ourselves that yes, humidity is probably playing an important role. So once we did this, we asked the question, okay, so what happens if we look at the spatial dynamics? So this is, again, observed is in green, simulated is in yellow. And we can see that it captures the seasonal pattern well, which I just mentioned, but it also kind of captures the spatial variability across Bangladesh pretty well as well. So center of gravity is giving me a measure of when the peak timing happens, and we can capture this longitudinal and latitudinal gradient as well. So then we took this and we said, okay, well, what happens in the future? So on the left here, I'm showing the baseline, which is current conditions, and then projected conditions using CMEG6, which is the latest climate models. We're using the ensemble model. And you can see that it's projected to get, it's not a surprise to anyone. It's also projected to get more humid. So the humidity curve shifts up as well. And the interesting thing to us is when we then put this into our simulation model, we see pretty different results for different parts of the country. And in some places we see burden not changing very much at all. So City, for example, there's not much difference. In some places like Select, we see the shape of the outbreak changing as well as kind of the burden. And a lot of this has to do with the fact that Sylhet is a part of the country that historically was never flooded, didn't really see a lot of monsoon rains, but it is projected to get much more humid in the coming years. And so it's moving into this kind of high risk transmission range for flu. And then in other places like Orissa and Xinjiang, we see a decrease. And a lot of that is being driven by the fact that these places, the temperature is getting almost too hot for flu transmission. And so it's, even though humidity is increasing in these places, we see that it's being offset by the fact that temperature increases means that it's too hot for, in our model, it's too hot for the transmission to happen. And so one of the things that we took out of this is that it's really, even in a small country like Bangladesh, I think it's really important to think about the variability across places and also how different predictions for example, precipitation and temperature might be playing off of each other to lead to kind of these unexpected results. Any questions? Good. Okay. So in other work, Rachel and I, along with Jess Metcalf and colleagues, have also shown similar effects for other respiratory pathogens. And we, this is a paper that Rachel and I first wrote when we were both PhD students with Jess at Princeton, looking at the impact of chickenpox. We see pretty similar relationship with humidity. And then we did a follow-up paper looking at RSV, and we also see pretty similar relationship with humidity. So we think that there might be for respiratory pathogens which haven't typically been studied as well in the climate literature. I think we're trying to make the argument that it's probably not going to have as big an impact as it will for vector and waterborne pathogens, but it's probably something we need to kind of think about, especially given that we see that the biggest change that we see of climate is not just on the magnitude, but also in the timing. As we think about vaccination timing and things like that, or think planning for hospital burden, we might want to think about changes in climate for the future. Okay, so I'm going to talk, how much time do I have? It's 10. Okay. Okay, great. So I'll talk about some much more recent work. This is very much more work in progress, thinking about not just climate, but the joint effect of climate and demographic changes. So there's many ways in which demography is, human demography is changing, and we might think about how that change might affect infectious diseases. One aspect of this that I worked a lot on is thinking about human mobility at the global scale. So this is looking at air travel in 1933 versus 2020. I think with COVID-19, there's not really any need to convince people that this is that this increased connectivity is problematic. But it's just the sheer volume of travel and connectedness and the rapid increase in this over time has been really at a scale that we haven't quite seen in migration studies and migration work. And we might also think about how climate is affecting population movement. So this is a nice paper by Su et al. That looks at IDPs are internally displaced persons. And so climate itself is having effects on where people move to, how people are displaced, and thinking about the types of weather events that can cause this. So floods and storms lead to the vast majority of the internally displaced persons. And often when we think about cyclones and hurricanes, there's the initial mortality impact, but then there's often these follow-on impacts that we see, for example, cholera outbreaks and so on. And so a lot of interesting and exciting work in this area, I think, is thinking about how these events lead to displacement, which then leads to these other health effects down the line. And then there's also urbanization. We might think about the many ways in which urbanization affects infectious diseases. So some of which is affecting things like, how the vector, what are the vectors that thrive in urban areas. So for things like dengue, chikungunya, Zika, these are all spread by mosquito that's pretty well adapted for urban environments. We might see less malaria. So, urbanization comes with many kinds of changes. And so some of these are positive, some of these are negative, both in terms of human health, but other indicators like socioeconomic indicators. And so, for example, we see less malaria in urban areas, and there's also, some work that's been done to show sort of income effects in urban areas. So urban areas in demography, because when demographers used to study this historically, urban areas is where you see the highest mortality, the kind of worst living conditions, and that's sort of really shifted to where urban areas, people in general do tend to lead kind of better lives and have higher incomes. And so this shift has also been sort of studied in the demographic literature. But in terms of respiratory pathogens, there's been some really interesting work by Ben Dale and colleagues looking at what do respiratory pathogens look like in urban areas versus non-urban areas. And they find that you have these more persistent outbreaks in urban settings, a lot of which has to do with your contact networks and connectivity in these urban areas. We've done some work looking at urban areas and how they act as amplifiers for outbreaks. So we wrote a paper on the chikungunya outbreak in Bangladesh, looking at how Dhaka city acted as the source that when you had the major holidays in Bangladesh, the outbreak spread really rapidly out of Dhaka city into many other parts of the country. And then more recently, again with Rachel Baker, we've done some work thinking about how urban contact patterns and daycare use and contact patterns in daycare can drive RSV dynamics in urban settings, which look pretty different from RSV dynamics in rural settings. Okay, but the part that I am probably most excited about in my work right now is thinking about aging populations and what that might mean for the burden of respiratory pathogens. So this is a figure from our world in data taken using UN data, looking at fertility rates across the world. So going from 1950 and then projected all the way up to 2100, and we see this very dramatic convergence of fertility rates globally. So roughly converging around 2.1, which is what we call replacement level fertility. So on average, how many children women would need to have in order for the population to stay roughly constant. So that's 2.1. Many countries are now below that. But it's seen this sort of dramatic convergence in the last 50 years or so in fertility rates globally. And what this means is that the share of population in different age groups is changing. And so if you look at the population share that's age 65 plus in orange highlighted here, it used to be at the lowest share in the population. And this was true actually for most of human history. This has been true. And then we kind of see this shift starting to happen in around 2020, where we see age 65 plus share going up in the population. And this the important thing for infectious diseases is it's not just the share of people in the 65 age group that's going up, it's also the number of people. Because we have population momentum, these previously large birth cohorts are now aging, we have both higher proportion and higher number in these age groups. And so we might think about how this affects infectious diseases. This is a really nice review paper in PLOS Pathogens, where Sorcy and Fib, they looked at data for many different pathogens. So the titles are too small to read, but this has things like there's SARS on the fourth one at the very end. COVID is here. COVID-19 is the third one. Flu is somewhere up here as well. So many different pathogens, but we do see that for most infectious diseases, it's the really young that we care about in terms of mortality, and it's the very old where it has risk of mortality. And so with demographic change, with falling fertility, and with population aging, both of these groups, the number and proportion of these groups are changing. And so we might want to think about what this means for infectious diseases. So one of my students, Callie Hoffman, and Rachel Baker and I teamed up to try and think about, okay, well, could we take a simple SNR model and then try and jointly model the impact of both climate and demographic change? So we took the simple classic SNR model, we just split it up into different age groups, and we said, okay, well, what are the ways in which we think the transmission curve might be affected? How do we think aging affects susceptibility, birth rates affecting pool susceptible people? Again, a very simplified picture of what happens in reality, but sort of helps us grasp that if we change any of these parameters, how much difference do we see? And so we can use this to simulate a flu dynamic. So this is a very hypothetical simulation. We're saying, let's just take today's age structure in the US, roughly the R0 that we think flu has in the US today, and let's simulate it. And here I'm showing that simulation for different age groups. So we have time on the x-axis and the outbreak curve for each age group. And you can see we have on the left, the age pyramid. So the orange is our current age pyramid in the US, the green is our projected. So you can see the numbers are increasing, and then the distribution looks quite different. And then we can think about, okay, if we take these projected age structures and projected climate conditions, what happens to flu in the US? So I think, again, this is a very hypothetical question. There's nothing about vaccination in here. There's nothing about immunity. And so, it's just in the way we phrase it in this paper is a thought experiment to think about, just to help us think about how do we think about these joint effects of climate and demography. And the joint effects that we see is quite interesting. So climate is changing the timing of the outbreaks, but the US is projected to get more humid. What that means is that you then move into the low transmission range of humidity. So it doesn't get as humid as Bangladesh. So you don't move onto the other side of the U-shaped curve, you're kind of in the low transmission zone. And so if we were just looking at climate effects, one might say, okay, well, it seems like climate might actually make conditions more humid, and so maybe you'd see more flu, or see less flu. But what we see as well, you see a little bit less, but really it's just shifting the timing. So it's shifting when the cases happen. And then if you layer on aging with it, well, we see fewer cases in the younger age groups, but then we see this shift in both timing and burden in the older age groups, and just by the fact that there's simply more people in these age groups. So I think even without simulation, we could have probably guessed this would happen, but I think it's important to think about how these climate and demographic changes might work in different directions, and it might give you a sort of unexpected effects. Okay, I'll talk about one last thing about aging, and then I'll take questions. So this is some new work, again, led by Kylie, a PhD student with us, and this is thinking about aging from another perspective, which is immune history and immune exposure. So this is a heat map showing flu seasons on the x-axis and age on the y-axis. So this is the typical Lexis diagram that demographers use, so cohorts, they move along the diagonal. So if you're interested in a particular time period, you look vertically in that period. If you're interested in a particular age group, you look horizontally across, and then cohorts go diagonally across. And then what Kylie did is she said, okay, well, let's look at all influenza mortality in the US. So she looked through all the death records available in the US from 1966 onwards, and looked for the ICD-10 code for flu and flu-like illnesses to see. And when we started off this project, we were just interested in understanding who is dying of influenza in the US. Are they in specific age groups? Are they in specific locations? What are the characteristics of people dying? And so what we did is we just took these flu mortality data and we fit a pretty simple mortality model. So Lee Carter model is a very common model used in demography to project mortality dynamics. But what it does is it has an age profile. So you kind of take out the age effect, and then it has a period effect. So we know mortality fluctuates in general year to year and seasonally. So it kind of takes out that period effect and takes out that age effect. And then what we're plotting here is what's left behind. So this is the residual. How far is our observed data from that simple age and period standardized model? And we see this interesting pattern that we thought was quite striking. We see that there's some action happening before 2009. But really what we see is that post 2009 in every H1N1 year. So 2009 was the H1N1 swine flu pandemic, where a new H1N1 variant got introduced and has been co-circulating with H3N2 in the US. And we see that these younger age groups have higher than expected mortality, given their age group, compared to historical averages. And then we see the older age groups seem to actually have some protection. They have lower than expected mortality compared to what's happening historically with those age groups. And so Kylie did some really great work to look at, okay, well, what does flu circulation look like? And can we match birth cohorts to what flu strain they first experienced? So this is the idea of imprinting that's been in the literature. I think some of the early papers in the 1960s by Thomas Francis coined the term original antigenic sin. This idea that what you are, the strain of flu that you're first exposed to in childhood, you have the highest antibody titers to that strain. And so later in life, if you see strains that are pretty different, you might have a worse effect, and if you see strains that are similar, you might actually have some protective effect. So this is what flu circulation looks like in the US over time. From 1918, that's the Spanish flu, it was H1N1 for a long time. So those cohorts that were born in that time period were probably only introduced to H1N1 and had repeated H1N1 infections for much of their young childhood and young adult life. Then H2N2 gets introduced. And then we have this shift from these group 1. So there's two phylogenetic groups, these are H1N1 and H2N2 are in group 1. We see this shift to group 2 viruses, H3N2 viruses getting introduced, and then 2009 with the swine flu pandemic, H1N1 and H3N2 have been co-circulating. And what we did is look at, okay, how similar were the 1918 H1N1 viruses to what gets introduced in 2009. And it turns out that there's quite a lot of antigenic drift in the between 1918 and 1957, such that the H1N1 viruses evolved, and the earliest strains of H1N1, what we call H1N1 alpha in the paper, seems to be the most closely related to the 2009 pandemic. And so if we put this together, let me skip to this part. If we put this together and we look at cohorts, and this is just the raw data, no models, we're just looking at age on the x-axis blue death rate on the y-axis. I've just arranged the colors by what strain people cohorts were imprinted to. And you can see these H3 and 2 cohorts. These are the youngest cohorts born roughly in the 1960s and onwards. These younger cohorts have experienced higher age-specific mortality at almost every age compared to older cohorts. And what's kind of striking about this, at least in demography, is this is really unusual in demography. In general, younger cohorts, we see improvements in mortality no matter what indicator you look at. So like HIV is a classic exception to this rule. I think young men after the collapse of the Soviet Union is the other exception to this rule. But in general, we see mortality improvements as cohorts for more recent cohorts. But what we've been seeing is that partly what we think because of this imprinting effect, we see that in H1N1 pandemic, there's these younger cohorts, the purple color right at the top, these are the youngest cohorts, actually have the highest age-adjusted mortality. And so the question that we wanted to ask is, okay, well, what happens if we simulate this and we say, what happens when these cohorts age? So these cohorts are now at not the high-risk ages for influenza, right? But these cohorts are going to age into the high-risk 60+ category for influenza. And we can simulate this. And when we do this, we see if we look just 20 years in the future, when these H3 and 2 imprinted cohorts age, we actually project a much higher burden by the fact of not just population aging, but this interaction of aging with immune history and early life exposure. So just to conclude, I think hopefully I've kind of convinced you it's a complicated story, and it's really hard to kind of tease apart all these different elements that affect sort of the burden, the seasonality, the geographic distribution, and there's many factors that I haven't even talked about here, right? So we can think about the immune history, we can think about demographic change, but there's also sort of societal changes, political changes, and many other things, and I haven't really touched on vaccination at all, but we think changes in how people, the types of vaccine that will be available, or maybe changes in vaccine adoption are also going to have an impact on this. And then I think what is also sometimes missing in this literature and sort of this mathematical literature is thinking about heterogeneity and impacts and who is most likely to be impacted by these changes. So aging and population aging is one aspect of this, but if we think about groups in the US that are currently most impacted, so if you look at flu mortality in the US, it is in every season disproportionately higher in Blacks and Hispanics. And so we might want to think about which groups might be most affected by these climatic and demographic changes. So thinking sort of like the more social science perspective, this fundamental cause theory of disease and who is at risk, I think will be more and more important to think about how do we put those things into these mathematical modeling frameworks to think about the future. So I'm gonna end by thanking my funding sources. Most of the newer work is funded by NIH. And then we have all our data and code if folks are interested. I also want to thank my group. They do all the hard work. And if anyone's interested in a postdoc, this is my advertisement for if you want to come work on any of these things, I'd be happy to chat. So thank you. Okay.
Thank you. Did that work now?
Do you want?
Now, okay, cool. Thank you. That was great. I have a few questions, but I don't want to dominate. So people have questions. Yeah. If you want to do this,
we should get the questions.
Yes.
Okay. Hi. So when we're looking at the impacts of climate drivers on flu, for seasonal flu, were the effects of relative humidity, you know, you're looking at mainly temperature and there's a lot of variation, but do you think that operating as main effects, or do you think the interactions between temperature and humidity, especially in areas of the world with that experience more variation?
Yeah, that's a great question. I think there are interactions, and I think some of the lab studies suggest that there's possibly some interactions. It was really hard in the Bangladesh study. So we did have an interaction term, but it's hard, even if there's an effect, it was hard to pick it up. So I don't have a great answer for your question, other than I think doing a similar model with data from other parts of the country, but or other parts of the world, with greater variation in both humidity and temperature might be the way to answer that. But that's a really good question.
And I also wanted to just ask about extreme events, and if you consider how that could potentially impact flu transmission, especially in the high transmission periods.
Yeah, that's a great question. I haven't worked on it directly, but I think that the sort of indirect work that can be done on it is sort of thinking about how, so I've done some work estimating mortality after cyclones and estimating displacement after cyclones. I think the cyclones driving population density and concentration and displacement is probably I would be shocked if it didn't affect transmission of any pathogen, including flu. And so there's probably maybe non-human behavioral aspects as well. But the part that I probably have thought most about is the fact that cyclone-induced displacement and this concentration that you see after cyclones and population density is probably.
Hello, thank you very much for this very interesting talk. I'm not sure if this might be maybe a little beyond the scope of your work, but are you able to, or have you considered including some sort of cumulative disease burden prior to the mortality? In, I guess, thinking of it, does that potentially like move that mortality window much earlier if there's an increased cumulative disease burden across that life course?
Yeah, so you're thinking about like a cohort that sees much higher burden throughout their life course versus cohorts that don't. Yeah, I mean it's a good question. I think our cohorts don't look that different in terms of cumulative exposure. So we do control for the intensity of the flu season, because that varies a lot. Your H3N2 years in the US at least tends to be much more deadly. And and risk of severity, like you were saying, more than anything else. I mean, there's some work on climate and COVID, but I think even that suggests that it's primarily right now. I mean, there might be a climate effect, but it might again washed out by the fact that it's just being driven by susceptibility and risk of severity.
So, a wonderful talk, one of my favorites of the fact. one of my favorites of the whole 20 years we've been doing this. Do you have an age limit on postdocs? 81-year-olds. I'm going to sort of with a teaser on a thing I'm interested. Could you include limitation and for example, your different flu strains? Did you treat them as just different bugs, or did you think of acquired immunity to one strain affecting transmission of other strains?
Yeah, I think the latter. And I think, I think our work might not be the best at speaking directly to this. But lots of folks have looked at antibody titers after, first exposure to H1N1 versus first exposure to H3N2. And I think the interesting thing is the original antigen sin was actually, sort of framed as like a negative effect. So it's, you would do, so if you saw many years of H1N1, you would actually do worse in an H3N2 season. And we do see some of that. The H1N1 cohorts do really poorly in H3N2 seasons. It's a little bit hard to disentangle that from the age effects because they're the oldest cohorts in those seasons. But I think, I think of it in the way that you're phrasing it, that the strain that you're first exposed to and how antigenically similar or distant is it from what you might see subsequently.
So I have a comment based on my own work. So we looked to see if there was difference in susceptibility and actually using our regards of Nicaragua household transmission model, if it was modulated by the microbiome. And we have evidence that it is, and then it varied by between influenza A and influenza B. And when you were showing the differences by dryness and humidity, I have a hypothesis here. When it's very dry, there's got to be a different microbiome. And so that the dryness, it may not just be the contact during, so that low humidity actually may have a biological effect. I mean, we all know that your nose is dry, right? And then similarly at high humidity, it's maybe not be a person effect, but when there's high humidity, you're more likely to have viruses and small particles stay in the air longer. And so that at higher humidity, there's just, we know that people, like maybe 50% of people with influenza don't know they have it. And so they're they're capable of transmitting it. And so the suspension in the air of the viral particles might explain part of the difference.
Yeah, that is really, I think the microbiome thing is super interesting. That's which is why I'm glad I'm here. I think that's these new ideas. The virus discussion, I think Anise Lowen and the colleagues in their original guinea pig paper did look at this. They looked at the droplet size, and they looked at how long it was suspended. I think you're absolutely right. I think that is a factor.
And so, I mean, that would be something that would explain it, but that's actually, those are both testable hypotheses.
Did you see any testable hypotheses in terms of why low humidity was such a factor? Or just a population model that?
No, so our one, we're not testing any sort of biological mechanisms, but I think the lab studies have been really great. I mean, guinea pigs are not humans, but the aerosolized transmission happens similarly. So I think,
Hey, thank you for the very interesting talk. I was very interested in this aging and the mortality relationship. I was wondering if you think today, as we have seen an increase of chronic disease burden across the world population in many developing nations, we used to think the primary cause of disease, that's the infectious disease. Actually, the epidemiological translation has shown that our burden of chronic and infectious disease. So I was wondering if for modern this kind of relationship, chronic disease burden should be considered as one of the core components.
yes, absolutely. I think it's definitely playing a role. The one tiny, probably not satisfying thing we do in this paper to try and control for this is we run the exact same model for a whole bunch of other causes that are non-infectious, some of which are chronic diseases, to see if we just pick up the same pattern. Because if we did pick up the same pattern, then there's something else going on that explains just mortality in general and this cohort time period interaction. You see clear cohort effects, but you don't see this kind of cohort period interaction that you would, then be able to explain this. So we try to do this a little bit, maybe not convincingly enough, but that's a great point. Yeah, I think chronic diseases are definitely changing, and there's clear cohort effects that show up, but our paper wasn't focused on it. So we just said, oh, well, this kind of it's not showing the same pattern. So
another related to like the nutritional status, lifestyle change. We also see lifestyle change across the world, especially among younger population. So,
yeah, absolutely. I think all of these things, and when I teach my demography 101 class, we talk about the various ways in which the demographic transition happened and what we think caused the mortality decline. And there's some people who are like, this is all nutrition, some people are like, this is all public health, some people are like, this is something else. But it's probably a combination of all of those things. Yeah, and I think the sort of Lee Carter model approach we thought was great because it kind of, the cohort effects would show up so clearly along the diagonal, and they do for some diseases very clearly show up. And so, it's sort of one way to say, well, it's not just the cohort effect. If it was just a cohort effect, it would just we would see it clearly as diagonal. So, but yeah, probably in reality, many of these things are.
Any other questions? Well, let's thank Dr. Mooney for wonderful talk.